Hi all, my name is Olawale Salaudeen. It's my honor to introduce Dr. Stephen Boppart, who is the director of the Center of Optical Molecular Imaging at the Beckman Institute, the professor and Grainger Distinguished Chair of Engineering at the University of Illinois, and one of our new MBA faculty. Dr. Boppart's biophotonics imagery laboratory is focused on developing novel optical biomedical diagnostic and imaging technologies and translating them into clinical applications. Professor Boppart received his PhD in medical and electrical engineering from MIT, his MD from Harvard Medical School, and his residency training at the University of Illinois in internal medicine. Dr. Boppart has received numerous honors, including MIT's Top 100 Young Innovators Award, an NSF Career Award, the International Hans Sigris Prize in the field of diagnostic laser medicine, and many more. It's my pleasure to introduce Dr. Boppart, who will give a talk on ultra-parallel label-free optophysiology of dynamic neuroactivity and circuit connectivity. Thank you very much, and I'll hand it over to Dr. Boppart.
All right, well, thank you very much for that introduction, Olawale. Really a pleasure to be here. Really want to thank Martha and June for this invitation. It was last minute, but these opportunities to really speak to so many trainees as part of this program and maybe tell a bit of my story as well, because I started my entire academic professional career in neuroscience and here at the University of Illinois. Through undergraduate and then graduate research, and now 30 years later, I'm here speaking to many of you, and I feel like it is such a moment for inspiration. When the way I was inspired, that hopefully that there's some shared inspiration as well and enthusiasm. What I wanted to talk about today is really when I was thinking about putting together some slides was what sort of tools and technologies might be available for you as trainees, as researchers, as faculty, as others that are interested in really probing the neuroactivity, the connectivity, the neuroscience in many of these various systems. And much of my background, my lab here at the Beckman Institute develops optical imaging technologies, and with the goal of really translating those into either clinical applications as well as for biological discovery. And we think about different ways of probing, and that term label-free is important because we also think about ways of extracting as much information out from biological systems without having to perturb that system in some way with dyes or stains or other approaches. Now, ultra-parallel will also become evident as I start talking about how we do this. I want to point out one thing too. You'll see some graphics that throughout my talk, and this is done by extremely talented graduate students in my group, Rishi Iyer, and it's just amazing how he can visualize a lot of the science that he's looking at in neuroscience and really relay this in graphical form. So really a talented individual about the scientific communication and how to relay these complex topics. So I'm speaking to I guess a well-seasoned and informed crowd, but I'm just amazed at the brain. Every time I think about the brain, the anatomy, the connectivity.
I'm sorry, I'm not sure if we're sharing the screen here.
Okay, let me see that.
Back to Chrome, or no, sorry, it's in Zoom. The camera. Yeah. Or actually, no, click on there. Yeah, there. Share screen.
Okay.
And there.
And present.
Sorry about that.
No problem.
Okay.
Can people see that now? Hopefully.
Yes.
Okay. All right. So just thinking in terms of just the complexity here and the enormous task that we have to understand the structure that exists, as well as that may not. So now the slide advancer is not working.
It's not, okay. Did that,
Maybe
did that do it?
that did it, but
Okay.
maybe I'll have to leave that present.
Is that,
That's something new. So if we hide that, we lose our,
this is new to me too, I'm afraid.
all right.
Oh,
Okay. Well,
Are the arrow keys working?
okay, now it's,
Okay, sorry about that.
we'll see. Technology. So in addition to that structure, right, there's also the mind, right? The mind in terms of the function, the physiology that takes place, this connectivity and making sense of it all. And again, just fascinating and mind-boggling when I think about that. In the middle, I think of neuroscience, and I think of the technologies and the tools and our ability to try to integrate between the brain and the mind and understand this complexity. And so that's what's driven me as an engineer, as a scientist, to think about how we can develop the tools and the technologies that can probe in new ways and extract and understand all of this in a better way. Now, the challenge from an engineering point of view is something that I encountered 30 years ago as an undergrad and a grad student here. And in fact, I pulled this up. This was my very first scientific publication in my career back in '92. It was work that I did in developing flexible multi-electrode arrays that could be peeled off of a rigid substrate. And this is maybe some of the first flexible electronics. They were passive, but they were electrode arrays that were also perforated. And so we could grow neurons, we can place slices on here, you could wrap this around very convoluted brain structures. This was all done in Bruce Wheeler's laboratory here at Illinois, and he's gone on to many other places. I think he's still at UCSD, but it was really about trying to expand our ability through technology to be able to record this neural activity. And so this stuck in my mind and still drives me forward in terms of the research that we do, that the single point molecular pipettes to be able to, impale cells and tissues and make these electrical recordings are limited. The electronics in Faraday cages, the complexity that's required to kind of isolate from interference and reduce the noise, all to make that single measurement in a single cell. And so our early efforts were in these multi-electrode arrays. Those still exist today in many different forms. And it's the ability then to go from a single point to maybe 64 or 256 points spatially distributed across space and make these simultaneous measurements. Well, again, I hope everyone realizes that while this is fundamental technology that is highly enabling, they're limiting if we're trying to think about more of the connectivity or the activity that's taking place over much larger scales that extend far beyond kind of what these limited regions exist. And then, of course, there's an incredible amount of science that has gone into understanding these neural signals that we recover from arrays that are spatially distributed, how to understand the relationships between the signals on each electrode and relate that back to the neural activity and connectivity within neural systems. Now, in the last decade or so, there's also been advances optically to be able to address this limitation and many other challenges. So optogenetics has come about, optophysiology, stimulation and recording systems. And this is, again, truly revolutionized neuroscience and this ability then to go in and not only probe single neurons, but now to be able to make multiple neurons across different circuits and tissues optically responsive, and go in then with either a single laser beam or multiple light sources and go in and stimulate across spatially across them. Now, a lot of this still relies on fluorescent dyes or genetically expressed calcium indicators to be able to make those neurons of that tissue optically responsive. And also to read out, to be able to produce light when these cells and are firing and active. So it's still a somewhat invasive, type process to be able to enable this functionality. Well, some of the work, I'm not going to talk about this particular work from my group, but we started thinking about, rather than spending all this effort to modify the biology to make it optically responsive, could we actually just modulate the light source that we use to stimulate these optogenetic neurons, or even naturally occurring photoreceptors like in the eye, and be able to control neural activity just through the shaped light. And so this has been, we published this a couple of years ago. This actually crosses over into this realm of quantum biology, because by changing the light stimulus, and it's not just about amplitude or wavelength, this is really getting into the phase and the spectrum, and for instance, ultra short pulses that would deliver light in different ways to stimulate neurons. And what we found is by doing that, by modulating some of these optical properties of the light pulses, we can actually control the functions of channelrapsin modified neurons. And so I'll point you to that if that's something of interest to you. So our motivation really for a lot of this technology development is that this type of discerning these types of neuroactivity is still a holy grail for a lot of what we're trying to do in neuroscience. And again, historically, we've been trying to do this with single point or multi-electrode arrays and optogenetics, these calcium indicators for reading, and voltage sensitive dyes for reading out activity, has enabled this visualization of larger circuits. And but of course at the expense of those modifications. So our hypothesis for much of this work is, can we use optics in new ways and develop high-speed label-free technologies to sense the changes that are happening in neurons or neural tissue that indicate this activity. And we can sense that through optical scattering, scattering of the light, the polarization state of the light that comes back, and also phase-based imaging techniques. So with optics, we have the ability to look at not just the amplitude of the light, but phase. And that phase is a very sensitive indicator of tissue structure or changes. And can we use those to track the electrical activity in a very high parallel, ultra parallel, high throughput type of system? Well, what this is trying to do is move from this invasive electrophysiology to the label-based optophysiology, and ultimately that label-free optophysiology, being able to do this even in vivo without perturbable systems. Now, to do that, we need new optical sources to be able to stimulate in different ways. We need new types of optical systems, and we need new types of optical imaging and data analysis when we develop these optical tools. So this is all kind of part of that whole package. So what I want to do is I want to just kind of walk you through some of the technologies that we've developed in my biophotonics imaging lab over the years, and then kind of end with this, our solution to some of this ultra parallel type sensing. So the first is really thinking about how we can combine a single light source to do all this type of optical, not only optical stimulation, but optical recording out of these platforms that use optogenetics. We'll talk a little bit about the neural circuit connectivity algorithms that we've developed, because when you have access to these larger numbers of neurons and small circuits within your fields of view, how do you make sense of that with all that optical data, video data that's coming in? I'll talk a little bit about some of our work on fast autofluorescence lifetime imaging microscopy. So FLIM may be a technique that you might be familiar with. It's, we know about fluorescence, but the fluorescence that's emitted has a lifetime. And that lifetime will vary depending on the environment that fluorophore is located in, and some of the cell metabolism and other parameters. So by measuring that lifetime, we can again get a label-free sense, and we're going after the lifetime of autofluorescent molecules like NADPH or FAD. I'll talk a little bit about what we call computational adaptive optics for imaging the photoreceptors in the human retina. And maybe you've heard of adaptive optics. That started with astronomy, when we're trying to look at, finding the starlight, light coming from individual stars passes through our atmosphere, and the light gets aberrated because of our atmosphere. And so there's many different tricks. You can actually correct that wavefront and create a very sharp image then of the cell of the stars. Well, that actually has been relayed now over to microscopy. So as we try to image deep into tissue is aberrating for these optical beams, and we can correct that. Well, that takes a lot of hardware. We figured out a way to do this computationally and be able then to reconstruct the photoreceptor mosaic in the retina. And then finally, I wanted to focus on this fast, label-free, full field imaging of neuroactivity, and we call it SPOOF, OCM, and I'll explain that as we go along. So starting off with this first one, and the single source. So this idea that optogenetics is out there, but very much many of these systems will require you to optically stimulate that photoactivatable protein or molecule, and then also optically excite say a calcium indicator. So that requires some increased complexity to be able to do that. And so again, from an optics engineering point of view, we thought, well, why can't we combine those in different ways? So we have a system that we built that uses a fiber laser to pump what's called a photonic crystal fiber. And this is a fiber that has a lot of different structures in it. What happens is the light interacts in a nonlinear way and generates what's called supercontinuum. So it's a light source. It generates wavelengths over hundreds of nanometers. And when you do that, you then have a lot of different wavelengths that you can carve up and filter out. And then we can select certain regions of that supercontinuum for the stimulation. In this case, we're going after C1b1. That's also supercontinuum to stimulate or to excite the fluorescence, the autofluorescence that would be coming from the tissue. So this was set up in an optical system, and then this is pretty much a typical microscopy, home-built microscopy system. And then we had a 561 continuous wave laser for also exciting at C1b1 and comparing. So coupled with that optical system, we had to really design new software to begin to think about how we could view the various neurons in our field of view. We can identify those by number. This was, again, in a transgenic mouse, brain slice tissue. The mouse was transfected and with GCAMP and also expressed C1b1 and cherry. And then we could identify the different cells for stimulating, optically stimulating. And again, this is all optical stimulation and all optical recording with the same system. But what this allowed us to do then is we could program which cells we wanted to stimulate, maybe pairs of cells, combinations that have different time delays, and then we could read out the optical response of this circuit in our field of view. And here's just a couple of examples. Now, one thing I want to point out, and I don't know if there's any way to get rid of this box. Hope that doesn't stop at all. So one of the things to point out is we're using laser sources, optical pulses that are very short, and we're doing this to stimulate, these different fluorophores or molecules. And one of the things we can play tricks with is when we compress these pulses to be very short in time, the efficiency for this excitation of that fluorescence increases. So that's what's meant by uncompressed and compressed. We compress these pulses down, and that just means we really get, we enhance those very weak signals that are coming from the different tissues. So that's really an advantage that we get with this pulse compression. But a lot of the excitation we're doing here is what's called 2-photon excitation, using these ultra short pulses. That's very different from what we would consider single photon excitation with just a continuous wave laser. So when we start to see then these neurons, these various structures, again, the green channel, this green background is really the spontaneous activity that's taking place, this readout, and the red identified cells are these optogenetic C1b1 transfected cells. So it gives us targets for us to excite. Then we can go in and we really have this ability to select the different stimulation protocols. So we can do CW at different points, P1 through 3. So again, that's a continuous wave laser, single photon excitation, or we can go into 2-photon excitation with these optical pulses. And so we can see the different types of optical responses that are coming back. This is a change in fluorescence over the background fluorescence that we see. The little red bars indicate the stimulation. And so looking at comparisons between the CW excitation and this 2-photon excitation between these different neurons and within this tissue. Now, we notice a couple of different things. The 2-photon excitation, we have the ability to modulate and modify these pulses, but it's also a very different type of response because often these 2-photon effects are much more localized. I don't have an imaging here, but they tend to be localized, even on the submicron level in terms of where that excitation takes place. Now we lost that. All right.
I think the click is going to Zoom instead of PowerPoint. Yeah, it should be good now.
Okay, maybe it just took a while, but okay. So that was just an example of that representative system for this optical stimulation and optical recording. Now, when we start getting all this information, these images that come in, thinking about how this connectivity takes place. Well, this brings it into this idea of connectomics, right? The comprehensive maps of the connections within an organism's nervous system, the brain or the eye. And of course, a lot of this is done with MRI and at larger scales. But we wanted to think about how we can start to create these types of spatial temporal connectivity maps within our fields of view. And as I started to allude to, if we can identify these individual neurons, we can think about ways then to stimulate single ones, look at their response within their circuit, and then begin to, in an automated way, stimulate pairs or triplets and different timing of this excitation. And in a sense, you could have a system automatically characterize the circuit connectivity within your field of view just by doing this, and then applying the math, figure out how each neuron is connected to its neighbors in this circuit. So this has been one of our goals for doing this. Now, the best way to collect a lot of this data is with the video capture. So these are hippocampal neurons isolated from GCamp transgenic mice. We're just globally chemically stimulating with glutamate, and we're using single photon excitation of GCamp, collecting videos at about 23 hertz or so, and you can see the activity, the different flashing. And we capture all this video sequence to then feed into this algorithm. And so in this case, you can kind of see our sequence where we start imaging for about 10 minutes, we apply stimulus, and then we look at different time periods after that stimulus to collect this video data. So from this time-lapse data, we can feed this all into an algorithm that starts to analyze all those responses. So the video comes in, we really break that up into different frames. We segment and condition so we improve reduce the noise in the images. We identify where all those neurons are in that field of view. We look at the time interval, we look at the change in fluorescence over their background. And then we have different ways of analyzing. We can look at the time domain, the time difference between when one neuron fires relative to its others, and or we can sort spikes, the firing that takes place. If there's a certain threshold, we can look at the distance between those and the intervals between those spikes, and then finally the frequency analysis. So if we can similarly look at how often and what frequency the spiking and activity is taking place. Out of this comes a lot of data in terms of describing that activity within that field of view. And so with this, we can generate cellular firing maps, which is essentially the number of cells in the field of view versus time. And we can see the brightness, the more red is the more active that fluorescence exists. And so we can just use this to kind of look at the activity overall. And then we can start to look at connectivity plots. So what is the relationship between individual cells and other cells? Now this you'll see is symmetric about this diagonal. And that's because in this particular way, we can't tell if cell A is connected to or exciting B or B to A, but that'll come in later. And then finally, we can look at some of this other ways of looking at time varying signatures over time for this what we're calling connectivity between the different cells. And then in this mode, we can identify the cells or activity within this red box are very high interactions between the cells, and the purple box is lesser or low activity between pairs of cells. Again, trying to think about graphically how to make sense of all this data. This is a frequency dependent analysis. So now we have these coherence matrices between pairs of cells. This is the phase matrices. So when we do this type of frequency analysis, we think about the phase relationship between the signals, we can then tell was that signal indicating connectivity coming because of from A to B or B to A. And that's why this is no longer symmetric about that diagonal, because there's a difference between those. And then finally, we've looked at different wavelet transforms which kind of break down on different spatial scales, the connectivity between cells over time. And what I didn't really describe is that these columns represent the control before, immediately after this chemical stimulus, and then at 25 micromolars of that glutamate kind of before and after. And so hopefully you can start to see that there's differences globally in these connections and activity between these cells. So again, another tool, another methodology for analyzing this. This other one is using that FLIM, fluorescence lifetime imaging for neural metabolism. And FLIM has been a technique that's been out there for many years, but it's always been slow. And it's because this fluorescence lifetime happens over picoseconds or nanoseconds. That's very fast. And so what people traditionally have done is they've excited that fluorophore multiple times, and they've kind of time gated, and they built up what that decay might look like over nanoseconds. Well, since the technology in acquisition and GPU processing has gotten so much better, we can now directly sample that decay, that fluorescence lifetime and decay. And so we can develop the fast FLIM system, again, using this 2-photon multiphoton excitation. So what you're seeing here is really cells that have been stimulated, and then looking at this type of differential response between different cells and their fluorescence lifetime. And what we're finding is that while most reported FLIM studies have looked at many minutes out after excitation, we're starting to see differences in individual cells and neurons, very quickly, just within, the immediately after stimulation, for example. And we think this is going to lead us to some more new information. But we wanted to apply this very sensitive FLIM technique to some of these long-standing hypotheses, this astrocyte neuron lactate shuttle hypothesis, that the role that astrocytes might play in supporting and providing energy resources to neurons that are firing. And with this ability to look at FLIM and as an analog to metabolism, we wanted to look at this further. So this has been a fairly controversial theory. There's some major papers, and maybe I'm even missing some others here too, that have debated this hypothesis, and we thought we can apply our tools to do this. So with this FLIM, we're actually getting at the metabolic activity of cells. And we did this metabolic imaging on GCAMP transfected hippocampal neuron cultures. We are looking at imaging not only that response via GCAMP fluorescence, but we are also then exciting the 2-photon excitation of that autofluorophore NADH. So again, getting this type of metabolic information from two sources. What we found, and I think it's well understood, that very often with this approach, we tend to see in resting conditions that these astrocytes kind of light up, are more metabolically active, and in fact, it's difficult to find some of the neurons because they don't appear metabolically active, at rest under these types of imaging conditions. So that the neurons have a very weak NADH signature, and those astrocytes tend to have the brighter signatures. Now, if we go in and start to stimulate, again, we have this multimodal 2-photon excitation of NADH, and also then the GCamp imaging of these cultures, and globally stimulated with glutamate. We immediately look for these differences in trends between the different cell types for these markers in metabolism. So if we look at neurons, for instance, with these excited conditions, we immediately see this kind of drop in this fluorescence. And with astrocytes, we tend to see this increase in fluorescence from these various parameters, suggesting there are differences, right, between the two cell types with this excitation, between metabolic activity differences between those cell types. With this fast imaging now, we can start to look at the responses over time. And what you're seeing here is in this culture, we've identified different cell bodies. The red trace is the 2-photon excited intensity. So that's the NADH. And so the black is kind of the mean lifetime, fluorescence lifetime of that fluorophore. The green bars are the periods of the optical stimulation. And we start to see some correlations between all this. We see some general trends that 2-photon intensity, again, indicating metabolism, tends to increase in some cells. It's flat, there's changes, but a lot of information we're still trying to back out and understand. Now, that's with optical stimulation. We can go in and chemically stimulate again with glutamate and start to look at over time what some of these changes are. And so this is really just a more dynamic set of information like I showed you before, but we see very distinct differences in the metabolism from these different cells and lifetimes. So overall, the major findings of this type of work was when we combine these imaging systems, we're starting to see that there really is this strong cellular basis for this different response between astrocytes and neurons supporting this ANLS hypothesis. And so, with these wide field type imaging, we can start to look at individual neurons and begin to probe a little bit more in terms of how the metabolism is shifting between these different cell types. Okay. So moving on to computational adaptive optics. The challenge here, of course, is OCT is a technique that we've used, optical coherence tomography. It's a technology that is the optical analog to ultrasound, but we send in light and we collect reflections and we can get depth resolve images of different structures, label-free, just based on their scattering properties. Now, OCT was developed at MIT in Jun Fujimoto's lab. That's where I did my PhD, and I was part of that when we started. It really has made its biggest impact in ophthalmology, biggest clinical impact. But the problem with imaging through the human eye is that the eye is very aberrating. And again, it's really amazing that our retina and the neural processing kind of sorts all that out, and we can still see, so well. But for us to be able to see with such high resolution the retina, to be able to look at the photoreceptor mosaic through all these aberrating structures, we had to figure out a way to do that. And so this is just a schematic of our OCT system. It's based on interferometry to collect those reflections that come back. And it's really set up to do two things, either a depth resolved collection so you can see different retinal layers, or this en face mosaic. And so with this technique, we're able to reconstruct what that in focus image would look like. So we get a typical en face frame. This is the retina. This is a vessel, actually a retinal vessel. We get phase information, and from the phase information, we can reconstruct the amplitude structures and see those individual photoreceptors. Now, how we do this is actually computational, and this is enabled by, again, computational power to understand what the aberrations are and how the aberrations affect that wavefront of the optical beam as it passes through the tissue. And this is described by these Zernicke polynomials, and what this is showing is that we really want a flat wavefront coming in, but it gets distorted by say the vitreous and the cornea. Well, we can reverse that distortion by just correcting those aberrations. And that's really how we then move from these blurry images to really the sharp images of the cones, the rods and cones in the human retina. So this was quite, I guess, a heroic effort too, because as you imagine, the eye is constantly moving, right? Saccades and all this movement. So being able to capture this data, correct this phase information, and reconstruct really was a heroic effort by Nathan, a grad student in my group. Okay, well, that's just really a collection of different techniques that we've used over the years to look at neuroactivity. And I want to come back to that problem of how we can make the sampling, this ultra parallel type sampling of neuroactivity even better. And this brings in a lot of the work that Rishi Iyer has done, complemented by his wonderful art as well. So I mentioned spoof OCM. OCM is optical coherence microscopy. It's like that OCT technique I just described, but it's microscopy. So it's en face, very high resolution. And spoof stands for super fast, polarization sensitive, off axis, full field optical coherence microscopy. So from this, and we love acronyms. So it's fast, which allows for high throughput. It's sensitive to polarization of light. Light, the birefringence of tissue and the polarization of light are new contrast mechanisms we can take advantage of. And then, of course, full field, we're going to capture all this information at the same time instead of scanning a beam to collect that. And what are some of these optical signatures that we could then collect from neuroactivity? Well, there are conformational changes in ion channels that occur with activity, right? And these minute displacements happen in the plasma membrane. We can pick those displacements up. There's ion flux with the neuroactivity, and as those ions move around, there's a change in the refractive index that takes place in the tissue. And so this is also something that OCT and optical systems are sensitive to. Finally, there's a reorientation of the cytoskeleton near these channels, and this changes the birefringence of the cells and structures. Again, something we're sensitive to. So these are again label-free changes in the cells and tissue that we're picking up. And we're using a similar type of interferometer OCT type system. We have an optical source. There's a couple of beam splitters. What's important is that we keep track of two different states of the polarization of the light coming back. And we have a detector that collects reflections from the sample arm and this reference arm. So that's how we get, that's interferometry. And that's how we get phase information about the light in addition to the amplitude information. And so we start to get images that look like this, where this is based on the phase and sort of the degree of polarization, this retardation angle. And again, looks very different from what we're used to, but again, these are subtle changes that are taking place in the optical pathway. We kind of zoom in, and here's a bit more detail. We have a number, well, this is just that optical system I was describing. Our detector is really this incredibly fast camera. So it's a 2D camera, it's capturing a megapixel worth of data at 4,000 frames per second. And this isn't even state of the art. They're much, much faster. We're just limited on cost right now, that camera. But we've been able to characterize, and you can see what resolutions we're talking about. We're on the order of 4 micron resolution in the XY plane. And we capture raw images like this. This allows us then to, with this system, to take the 2-dimensional Fourier transform, and that helps to isolate the different components, the phase components of our signal. And then we can get amplitude and phase values for these different, for this field of view. And from that, begin to look at these sensitive changes. Now, there's a lot of challenges with this, right? We're getting a 200 by 200 micron field of view at 4,000 frames per second, roughly 1 micron transverse resolution. And we're calculating that birefringence. There's challenges. We have to make some assumptions in calculating this birefringence to make this fast and watch out for things that maybe get a zero in the denominator and make this type of analysis explode. But we're able to approximate that birefringence and do that. To test this, we started using NE4C cells, differentiated mouse neural ectodermal stem cells, and then we can actually differentiate these with retinoic acid and use that as our sample here. And what type of data we get now, if we look at this field of view, we can assign different regions of interest. And again, at every pixel. So we're getting all this information at a pixel by pixel level. And if we look over time, we're sensitive to displacements, to changes in these neurons on the order of just nanometers. And that comes from that phase sensitivity. So we can start to see these differences between regions of interest. And you can start, if you kind of blur your eyes a little bit or from that distance, you might even think that those are electrophysiology traces, but they're optophysiology traces occurring across, again, these millions of pixels. Okay, yeah, we can zoom in here and we can see over time. In this case, this is looking at birefringence. So that's the change in the polarization. This column here is looking at physical displacement. The vertical scale is 10 nanometers. And then the various regions of interest, the vertical dotted lines are periods where we optically stimulated this. And then we do have an electrode in here too, that we can electrically stimulate or electrically record for standard single point electrophysiology. So if we distill a lot of these down, we see there are certain regions that are far more active than others. If we just average many of these, we can start to see that after stimulation, these changes are taking place. But hopefully you appreciate there's a wealth of information and data here. So this neural activity that we're seeing can be expressed as both as phase change as well as a birefringence, and we're capturing these kilohertz frame rates all across this field of view. We did do some of these electrophysiology recordings just to validate when these changes are taking place and look at standard deviations of that activity. This is the optical activity, and these are just the electrophysiology recordings that are taking place. So you can start to draw some correlations between these, even single large spikes between them. So clearly there's a difference between rest and activity among these cells. But this really provides this ability for very high throughput, because now, again, we can look at it on a pixel by pixel level where this activity is taking place, and start to then collect all this data, feed it into these connectivity algorithms, and start to analyze how this optical activity or the optical measurements of neural activity is taking place on a very ultra parallel scale. So this was just another example where we used a different cell culture. These neurons had channel rhodopsin and YFP, so we could optically excite those, and then collect this data, wide field the collection as well, that fluorescence. So we started to correlate the fluorescent activity with the electrical activity, and in a very similar way, again, getting just large amounts of data. We can identify different regions that are particularly active, and again, these are optical traces, not electrical, but we can start to see that there's correlations between the two. So if we kind of zoom in, we can see again these familiar optical traces at different regions of interest. What we can come back to is that connectivity algorithm. So now we have tens of cells to start to look at that connectivity between them and use those same types of functional analysis to look at how the different cells within that field of view are connected. And again, this is just a comparison between the optical responses and the electrical recordings that are taking place in that culture. So, and kind of zooming in, where now at different time points, we can really back out how is what we're calling connectivity or activity between different pairs of neurons is taking place. So a lot more data to now analyze and review. So just to kind of wrap up where we're going with this particular technology, we really want to think about improving some of the fluorescence images. We're thinking about more convenient light sources, better cameras, a faster camera, 4,000 frames currently, and easily we can go to 10,000, 20,000 frames per second. Begin to correlate more with electrophysiological recordings. We think, you know, the correlations are strong enough that we are seeing these changes due to the electrical activity. And then there's just a wealth of other new optogenetic channels that we can look at. So these optical imaging technologies really provide a new toolkit, set of tools to elucidate these questions at the single neuron level and these neural circuits over our fields of view, and even, you know, as we're thinking about in the living human retina. And this is enabled by these fast label-free optical techniques. These computational algorithms are key too. And the computational power that you now have at your disposal is just, you know, incredible to think about, and what that can do for correcting aberrations, for analyzing this type of activity, connectivity. And again, these are still small fields of view, and we have got a lot more brain and tissues to cover. We contend so much of my work is focused on label-free, and I think that this allows you in a minimally perturbative way. You're still putting energy in for the light, but you're not really physically, you know, impaling cells with electrodes or disturbing that environment, or even genetically modifying the cells to enable to get this understanding. So we think that this has a lot of potential to rapidly move toward in vivo studies and even human studies as well, as shown with the human retina, for instance. Now we're at 40,000 samples, spatial samples at 4,000 hertz. So hopefully you appreciate the ultra parallel nature of this. And for me, 30 years later, to go from the single point electrode to this type of sampling with so much data, it's just mind-boggling. And we want to be able to use this type of optophysiology for many, many other applications. So I just want to thank a great team, group I have in the Biophotonics Imaging Lab. We have, you know, students at the undergraduate level, grad students, postdocs, medical students, a lot of collaborators, and a lot of clinical collaborators that are working together with us as well. So thank you for your attention. Happy to answer any questions you might have. Yes.
It's very wonderful talk. So I have two questions about SPOOF. I mean, my lab does very old style physiology. We're ion channel lab, so we need a single channel level structural function studies. But I really like the fact that we can do label-free imaging. So two questions. One, the first one is, you showed all the data from different points in your image, right? And this is a high density hippocampal neurons, or these cell lines that are differentiated to the neurons. So can you really know what those points were? Is it on the soma, axon, or dendrites? Because as you know, neurons are polarized cells.
Yeah.
Different neurons have different function. So can you distinguish that, or are you trying to push this technology forward to get to that?
Yeah.
And then the second question is, can you push this technology to really look at the waveform of action potential? Because just tonic firing of action potential versus burst firing of action potentials, and different ionic currents are responsible for switching from first of all tonic to burst firing, is very important information that we need to know. Because, for example, epilepsy that we are studying in my lab, burst firing is really one of the key features of the neural activity. So can you use the SPOOF label-free imaging to really distinguish those waves?
Yeah, good question. So I think the first question how are we confident of what we're seeing and where are we localizing these signals? I didn't show you some of the complementary images where we can get like a standard phase type image where we can identify the neurons, the cell body, the axons, and where those are all located. You can kind of get a sense of some of the structures, but yeah, it's difficult from those phase images. So those are correlated, and we can go back to those images so we can know exactly what, what part of the neuron or off the neuron we were. And very often we will sample off of any cell or structure to get that background signal too. And then the following question, I would predict that yes, we can kind of see that bursting behavior. There wasn't any examples, or maybe from what there may have been a couple of traces where you started to see like a bursting behavior from a particular region. But with our temporal resolution that we have, we should be able to pick all that up. And I think just looking at different situations where you can induce kind of that bursting and track that should be possible.
So have you tried pharmacological treatment? For example, if you can block potassium channels or even T-type calcium channels that are important for first priority. Have you then looked at the changes in your signal?
Yes, yeah. So that the last paper that Ravi just published this year in iScience, we were looking at tetrodotoxin, and we've been looking at other channel blockers, and we clearly can see the expected response, with all those traditional measures. So so I think that in fact there was another study that we just published that is looking at this phase information and trying to develop new models even for the mechanics and the effects of channel opening. So even at the single cell subcellular level, what sort of changes does that induce on this phase measurements that we're making? So that's a highly biophysics type paper, but one that I think suggests that we have access to that information, and it's just trying to sort it all out.
Yeah. Thank you.
Yeah, but
Fascinating talk, Steve. I'm just wondering, I'm not sure I completely understand the OCM and how you're using it, but I noticed that in your mappings, you had a scale bar of 25 nanometers. Are you looking at motions of the neurons?
yes,
Individual neurons?
Yes.
On that scale?
on that scale.
And how does that relate then to the firing?
Yeah. So with interferometry, OCT, we have this ability to get the complex value of that OCT signal. So we have amplitude and we have the phase. And again, that phase is a much more sensitive indicator of changes. And that phase can come from either a physical displacement, a movement of a scatterer, or it could come from a change in the refractive index. So if there's different ions or things that will change the refractive index of that volume, then that will appear too on this phase shift. In fact, Gabby Popescu built his entire career around quantitative phase imaging, going right after that, as a tribute to what his work has made. And so that phase is what we use, but we have the sensitivity of really picometer displacements and extremely sensitive. And so the nanometer displacements on those scale bars are real. Now the question all then becomes: is it movement or is it refractive index change? Right? And even Gabby, the whole quantitative phase group struggles with that. You really can't differentiate that just by looking at that phase change, a direct one. So we suspect it's movement, and we know that swelling of axons occurs with electrical activity, and there is this the birefringence change. So a lot of the cytostructural elements, or as you can imagine, as like an axon will swell, there's going to be kind of a structural rearrangement of the membrane itself, and that will change the birefringence.
But those would be over very long time scales relative to what you're.